All right, let's take a look at the nephron. This is obviously zoomed in to really just one nephron. There are millions of these things in the kidney. And what I want you to see here, first of all, is just the interaction between these two sets of tubes. The first set of tubes, like we saw before, is all about blood. And you can see here that we've got blood coming in, forming this glomerulus, which is nothing more than this really fine set of tubes or capillaries that have little holes in it. And that's going to allow certain things to move from the blood and into this second set of tubes, or the start of the second set of tubes, called the Bowman's capsule. But sticking with the blood, once it leaves the glomerulus, you can kind of see that these capillaries continue to sort of branch off and they sort of drape themselves around the nephron. And again, just imagine this happening with the millions of nephrons that are in the kidney. And then they return and reconnect back up and form the vein that then goes out as the renal vein and back out of the kidney. All right, the second set of tubes, like we said before, starts here as the Bowman's capsule and then becomes this tube here. This is the nephron now, but this is the proximal tubule and then snakes down through here. Now, the stuff that's traveling through here is called the filtrate. This is the stuff that has been literally sort of pressure filtered out of the blood. And as it travels through this tube, the proximal tubule, and then down the loop of Henle, back up, and through the distal tubule, and through the eventually into the collecting duct, different things will be sort of pumped in, pulled out, and whatever you're left with inside of this tubule is basically destined to leave the kidney as urine. This collecting duct here will join up with other collecting ducts and then empty into the renal pelvis of the kidney, and that renal pelvis will then empty out its contents, the urine, basically, into the ureter and into the bladder and out of the body. So that's the two sets of tubes that we're talking about here. All right, let's talk specifically about what's happening inside here. This is where it all sort of starts. When we see this, and I'm going to try to make this large enough so you can kind of, hopefully, see it, and it'll pop out. I'm going to draw using two different sets of colors here. The first set of colors I'm going to use is to try to show things that will be pulled out, let's say, actively. This is through the use of pumps, and that's going to require ATP. And I'm going to try to draw that using, let's red is what I'm going to use. So in this, before we get into that, we're going to say that this is under pressure. This is going to actually sort of push out here certain contents, and I'm going to draw an arrow here like that, and maybe that's not good enough to see. Let me just backtrack this a little bit. Let's go a little bit larger. All right, so into here, from the blood into the Bowman's capsule, you're going to get things like glucose. You're going to get certain ions like sodium, potassium. You're going to get chlorine ions, amino acids. You will have stuff like water and other things that the body doesn't need. It could be certain types of chemicals like you get from different types of medicines, medications, and stuff like that that are dissolved again into the body and are small enough to pass through these tiny holes in the glomerulus. And I don't know if you can see that here. I'm trying to draw these little tiny sort of dots representing these tiny holes that will allow small enough particles that are small enough to actually pass through. That means that things like red blood cells, proteins, those will not pass through because they're just simply too large. Those will just stay in the blood. And those things, when they do stay in the blood, those will just sort of continue on and move through the capillaries and out of the kidney through the renal vein. All right? So, like I said before, the things that will pass through and into the Bowman's capsule and into the start of this nephron here will be stuff like glucose, amino acids, sodium ions, chlorine ions, potassium ions, water, and other things that the body doesn't want that are small enough to pass through. Now, let's move it to here. We're now going to move into the proximal tubule, and you can kind of see the proximal tubule is this first sort of coil of tubes that we see here. We are going to pump things out because there are things that made it into the filtrate, the fluid that's in the nephron here, that really is going to leave the body. unless the nephron pumps it back out of the filtrate and allows the blood to take it back up again. Because there are things like glucose that we're going to want. Amino acids we're going to want because we have uses for those things. We don't want to just sort of let that out in the urine. That's just going to be a big waste. Same thing with sodium and potassium ions. We're going to have a need for those things. So here, using the color red, I'm going to try to sort of show that this is where those things are actively pumped out. I'm not going to write it down there, but those arrows represent the active pumping out of stuff like glucose, sodium ions, potassium ions. And when that happens, all right, maybe I will try to draw it in there. Hang on a sec. Bear with me here. So let's see. We're going to have, this is going to be like glucose. Ooh, that was not an L. And that is not red. Sorry, everybody. Glucose. We're going to have amino acids. I'm going to put that as A dot A dot. We're going to have stuff like sodium ions. Those are going to be actively pumped out. Same with potassium ions. Potassium ions. All right, those are all going to be pumped out actively. Now, when those get pumped out actively, you set up a sort of like a gradient here. You set up this very sort of high concentration of these, especially these ions, but also with amino acids, glucose. And what ends up happening, other things get sort of pulled along with it passively through either osmosis, if we're talking about water, or just simple diffusion. Because or not diffusion, but things that pass through passively because they get attracted to some of the other things that are leaving the nephron. So I just said water. Water's going to come out, and water I'm going to draw again because it's passive. I'm going to use the color blue for that. So let's draw water coming out there. So water is coming out. All right. And so are things like chlorine ions. But let's talk about water first. Water's going to come out because of osmosis. And because you have this sort of high concentration of stuff like sodium ions, potassium ions, you sort of create this very hypertonic type of condition outside of the nephron. And so as a result, the fluid that's inside the nephron sort of has this hypotonic type of relationship with it, and the water's going to want to try to balance that out to reach isotonicity, and it's as a result going to pull out the ions that way. The water that way. So let's just draw in the label water. All right, so I'm going to draw in H2O. And the other thing that comes out passively are stuff like chlorine ions. And the reason why, if you look at what else is being pulled out, we saw that sodium and potassium ions, which are positive cations, it's going to then sort of attract and sort of encourage the passive release of chlorine ions outside of the nephron as well. So these guys will come out, chlorine ions, for example. Now, are these the only things that sort of come out at this point? No, there are other things, but these are the things that we're going to focus in on because we've seen and we've dealt with these types of particles and compounds before. We've talked about glucose, we've talked about sodium, we've talked about chlorine, we've talked about water. So we're going to focus in on this because now we're getting a chance to see how your body is able to reclaim these things from the filtrate before it leaves the body as urine. All right, so that's sort of the first stage there in the proximal tubule. We've got some stuff moving in, we've got some stuff moving out. Oh, that's really small. All right. Now what we're going to do is have like we said, the filtrate continuing its way down through here. Now we're getting into this thing called the loop of Henle. And here we have the filtrate coming in through here. The water, again, this part of the loop, what we call the descending loop of Henle, is going to be permeable to water. So that means it's going to let water sort of move in and through this sort of membrane or this wall here And when it does allow water to move through, it's actually going to cause water to move out passively. So I'm going to draw that in again and just sort of draw that in as H2O. All right, water comes out, and there is a reason for that. We have here, again, another hypertonic type of situation outside of this loop of Henle here. It's very salty here. And we're sort of dipping into the renal medulla at this point of the kidney. It's salty in here, and there's a reason for that. But because it is salty outside, again, osmosis occurs and water leaves, again, this filtrate that is sort of traveling through this tube. When, for some reason, you move up and going in the ascending loop, or the ascending branch of this loop of Henle, it's also going to allow salt or sodium to pass through. It's not going to allow water to pass through. So this is where it sort of becomes water sort of resistant, or if you want to say it's sealed against water. It is going to allow stuff like sodium to pass through or to leave passively, because again, it's going to try to, through diffusion, balance out. The reason behind this is because as the filtrate passes down through here, when this filtrate loses water, then it sort of becomes a little hypertonic in here, and so as a result, the salt is going to move, or the sodium ions, I should say, is going to move out, because here, at this point, water can't sort of move back in to balance it out. So it's kind of this weird trick it does. Here, water can leave, and when it does, this filtrate becomes sort of hypertonic a little bit, and as a result, when it comes to this part, this is the part of the nephron that we said, well, water can't pass in and out anymore, so instead of water being allowed to sort of pass back in to balance it, the only thing that's allowed to pass are these sodium ions, so if water can't pass through and back into the nephron, sodium's just simply going to leave. And that's kind of the relationship there. But it doesn't answer the question why the medulla is so salty. Why is it so salty in this part down here? Well, that's answered when we get to this part of the loop of Henle. It is here where active transport of sodium occurs. I'm going to do that. That's supposed to be an Na. Sorry, everybody. Na is actively pumped out. And because that happens, similar to what we saw in the proximal tubule, other ions sort of passively come out as well. And one of those other ions that gets passively diffuses out, or gets attracted along with it, is chlorine ions. All right, because again, the chlorine ion is a negative ion, and because the sodium's actively getting pumped out, it's a positive ion. Chlorine just sort of gets attracted to this movement because it is attracted to those positive charges, and it sort of follows passively out of the nephron, and yeah, through just attraction to this positive ion. The other ion that also gets sort of pulled out passively is potassium. And again, it's this diffusion. As these particles start to move out, and the water in particular starts to get pulled out of this nephron, similar to what we saw with the sodium here, potassium does the same thing too. It sort of starts to react to the fact that there's a higher concentration of potassium inside of this nephron now, and it's now going to want to sort of balance that. So it's just passively going to come out through diffusion. But this helps explain, this active transport of sodium helps to explain why the medulla is so salty, all right? And that's going to then encourage, again, more water to come out as more filtrate passes through this loop of Henle here. So that is the loop of Henle. Next, the filtrate continues on up and into this thing called the distal tubule. So this part all through here, like this kind of snake of tubes here. It's here where other things are actively and passively pulled in and pulled out of the nephron. But there's a unique thing to this. This is now under hormone control. So this is where the fine-tuning of the filtrate can take place. And really, what's going to control this is going to be dictated by the conditions of the body. And that will be detected through these sensory sort of receptors, and this control center of the brain, the hypothalamus, will then cause the release of hormones via the pituitary gland, and one of the hormones is ADH, or the antidiuretic hormone, and that will have a direct impact on this part of the nephron, the distal tubule, and also the collecting duct. It will, and other hormones will cause the active uptake of ions like, whoa, that's a big arrow, sodium. So let's draw in sodium here. Sorry, I have to sort of scroll in and scroll out because I'm just trying to do this on this tablet screen here. So sodium is going to get actively pumped out, for example. And again, depending on the conditions of the body, this might happen, this might not happen. But it's going to be determined through hormone control. Because sodium is leaving, this will also encourage the passive movement of other things, and you can guess as to what those things could be. You would have stuff like chlorine ions passively leaving. So let's see, chlorine ions passively leave. Again, because those positive sodium ions are leaving, these negative ions are also going to want to leave as well. Another important negative ion, or negatively charged ion, is this bicarbonate ion here. That is going to also get pulled out. Now that's an important one. We've talked about that with the acid buffer system, the pH levels that we can control and balance out through this bicarbonate buffer system. This is the stuff that we need. So as a result, this is a nice way to be able to reclaim that and prevent it from leaving the body as urine, because we might need some more of that. The other thing is water. And water is just simply going to go simply because of osmosis. We've got this situation here again where we start to, with the pumping of sodium ions, get this sort of salty type of environment happening. And water, once it's allowed to leave, this again under hormonal control, it might not allow water to leave. It might make this part of the nephron impermeable to water, in which case it's not going to be allowed to leave. But let's say, yeah, the antidiuretic hormone is in play, and so as a result, it allows water to leave this part of the nephron, this distal tubule, as well as the collecting duct, and water is allowed to leave through osmosis, and that's a great way to reclaim water as well. The last thing I want to talk about here is that we also have another type of active transport, but it's an active transport of an ion into the filtrate, and this would be hydrogen ions. You can see hydrogen ions being pushed or pumped actively into, I'm running out of space here, but let's just draw it here. Hydrogen ions could be pumped into the filtrate. And again, that would be, as an example, a way to help get rid of these hydrogen ions that have been produced as a result of the buffer system, to help support the buffer system. If the blood is too acidic and you want to swing the buffer system so that it's getting rid of those hydrogen ions, you're going to want to release those some way, and they're released through the urine. And that's done actively. All right, so that is sort of what happens there at the distal tubule. The last thing that I want to just sort of draw your attention to is, again, this passive movement of water through osmosis, again, in the collecting duct, which is determined again by hormones. When the hormone ADH acts upon this part of the nephron, the collecting duct here, all it's going to do is it's going to either make this part of the tube or the collecting duct permeable or unpermeable to water. If ADH is there, it's going to make this collecting duct permeable to water, so it's going to allow water to move through the walls of this collecting duct. And again, because this is a very salty area, we're getting down into the medulla again, it's going to have water sort of leave passively through osmosis. So this is not an active transport. This is still very much passive. But again, you're seeing here that hormones can control even the passive movement of materials in both the distal and the collecting duct areas of the nephron, simply by making it these, whether it's chlorine ions, water, or bicarbonate ions, allowing it to pass through the walls. And that is determined by hormones. That's kind of it. And I know it's a lot. The last thing I want to mention here before I forget is that notice this is again where the importance of these capillaries that are draped all over this nephron. This is again to allow for all of these things that are leaving, all these arrows pointing out of the nephron. Those aren't just going to hang around here. Those get sort of collected back up into the blood that is circulating through these capillaries and then leave this area through the vein there, and out through the renal vein back into the body. So again, just highlighting the importance of this network and mesh of capillaries. I think that's it. That is one long video, but again, people wanted to sort of know this in detail, so there it is. Good luck.